Welcome back to Genetics in the Paddock with Emily. Today is our last episode of Season 1 and it's a little bit different to usual. So today we'll be sitting down and doing a bit of a Q&A with a few of our guest speakers from Season 1. So I'm very lucky to be joined by David Johnston, Brad Walmsley, Sam Walkom and Tom Granleese. So thanks to everyone for taking some time out of your days to come and sit with me to film this podcast. I've got David, Brad and Sam sitting with me in person, and I've got Tom dialing in from Teams, and we have a whole range of questions that you guys have sent in. So I'm really excited to basically just jump straight in and see if we can work out some of the answers to these very interesting and diverse questions. So with that, let's get into it. So Sam, we had a question come in as well, and one that I think you might do really well at answering. So it is, how do commercial breeders benchmark their genetic level and what tools are available to do this?
Thanks, Emily. So yeah, good question. I'll answer it from a sheep industry point of view, but most of the theory aligns with cattle and other species. I think in sheep the easiest way to do it is to track a phenotypic mean of how your flock's performing. Data helps you understand what's going on. When we look at a phenotype, we do take into account that some of that variation across years will be due to the environment. And so taking note that in tougher years, you're probably going to have slightly lower phenotypic means. From a genetic point of view, if you're buying sires from ASBV studs, the easiest way to do is to take a mean of your sire team. This gives you an indication of the average genetics that is coming down through the sire line. So each generation should take on half of that, and that'll be your improvement. So if each year you're buying a sire team that is genetically superior to the last year, that'll give you an indication of roughly where you're trending genetically. In regards to sheep, there's other options. In the merino industry, there is a tool called a flock profile. This is done where we take a sample of the flock genomically, get the genotype, and then we use a back transformation out of the sheep genetics analysis to give us an estimation of the average breeding value for that flock genomically based on their genotype. There's a couple of projects on the go at the moment to expand that to becoming within flock individual tools, the genotype prediction in sheep. That will be available in merinos in the near future and then across the more non-merino breeds shortly after, hopefully. So that's probably a few ways in which we can make a progress, but easiest way is track your sire teams across years because they'll contribute most to your genetic gain.
Great, thanks Sam. Those are some very great practical tips, and I'm sure people can definitely take that on board to benchmark their genetic level. Thank you. All right, Tom, so I'm going to ask this question to you. So the question that came in was, given performance is often more driven by environmental and management factors, when should people start focusing on their genetics?
Well, that's a very good and curly question with a lot of grey area, Emily. The first thing I would say is if you're running an enterprise which has breeding, then there is always an opportunity to be focusing on genetics, because if you're breeding animals, then you're selecting animals, regardless if your management's at 30% of the optimum, 50, 90, 100% of the optimum. In saying that, if I was to be investing lots and lots of money in a total production system, I'd be wanting to really focus on getting the management of a property correct. So that's the health of the soil, health of the animals, and the genetics themselves. So, at the end of the day, if we have high performance genetics, there's no, if we liken it to a Formula One car, there's no point feeding that Formula One car diesel. So to extract the full genetic potential, we need our management right. And that's sort of the premise of a lot of the bred well, fed well workshops that are rolled around the country. So they do go hand in hand, and that, we need to be focusing. We always have an opportunity to focus on genetics from the get-go because we need particularly sires to breed our next generation. But yeah, in terms of total dollars invested, there's no point investing in high genetic merit or high performance genetics if our management can't support that system. Genetics is a permanent and ongoing thing, so you just can't chop and change your genetics like you would with a feed cart. So If there is environmental challenges often in your environment, then in your breeding objective you probably should be including some of those resilience traits added into your breeding objective. In saying that, you can have a top 1 percentile fat animal who has a body condition score 1. But just because they're genetically fat doesn't mean that you still need to feed for that genetic potential to be exhibited by the animal.
Great, that makes complete sense. Thanks, Tom. So, Brad, how would you manage traits that have unfavourable correlations in your breeding program design?
Most logical way to deal with them, Emily, is through using the most appropriate index. So each index has a genetic correlation matrix behind it, which contains all the genetic relationships between all the traits that are in the index. So the index is fully aware of what their relationships are, and then the index uses the economic evaluation to create economic values that put the right weighting on each of the traits given the relationships between them. So for a breeding program design, the most useful way to deal with traits with unfavourable correlations is to use the most appropriate index that fits your system. One good example is the relationship between growth and calving ease. In all our indexes that have calving ease EBVs used in them, the relationship between calving ease and birth weight is taken into account. The bigger the birth weight, the worse the calving ease, and then the relationships that birth weight has to the growth traits, bigger birth weight, bigger growth potential. Generally, that's all taken into account inside the genetic matrix as an example.
Okay, so can you just give a quick explanation for those who haven't really heard much about unfavourable correlations, what that exactly means?
Well, what that means, unfavourable means a correlation between two traits that's actually in the direction that you don't want to have it, to allow you to take the two traits in the desirable direction for each. So the example is growth. You want to increase growth, so increase weaning weight or 200-day weight, but you don't necessarily want to increase birth weight because a bigger birth weight is associated with a more difficult calving. So the two things, birth weight and weaning weight, have positive correlations, which means that if you increase one, the other would increase. So that's unfavourable in the sense you want to either maintain birth weight or shrink it a little bit and still increase weaning weight. So the positive relationship between those two traits is unfavourable in that breeding objective.
Okay, that definitely clears it up. And we had another question for our Q&A, which I'm assuming is fairly related to the one I just asked you. But the question is, how far can we push our animals genetically, and what is considered too much?
What is considered too much for a production trait is when that production trait negatively impacts other production traits to the point that profitability from those animals becomes reduced. So how far you can push a group of animals genetically is contingent on where they're starting. So if they're already highly productive, you may not have much room to push them further before you, with a given trait, for example growth, that you start to impact things like fertility or calving ease or carcass quality or something. So it depends where they're starting from, and you've gone too far once they start to negatively impact the other traits in the system. So an example might be pushing really hard for growth to the point you might nearly be single trait selecting for growth. What will happen if you don't monitor the cow size is she will dramatically increase her mature size, and then that has flow-on effects for on-farm feed costs, on-farm methane production, potentially issues around calving ease. So, you may push growth too far if you're not monitoring mature cow weight and holding it at the right level. So it depends where you're starting and what the ultimate trait is. There's not one single answer to that, unfortunately.
Okay, so that kind of goes back to the topic you were speaking about in the podcast about having a fairly well-rounded breeding objective that would cover.
Correct.
Cool.
Correct.
And going back to the breeding objective kind of topic, we had another question as well. So what new traits do you see becoming part of breeding objectives? I know we spoke a little bit about methane, but are there any others, or is methane the biggest one that you see being incorporated in the future?
In the current world, methane's a big one that's being talked about a lot, and particularly with MLA's CN30 target. But there are other things like traits around cow body composition and cow welfare in the production cycle. They're becoming more important, particularly the cow welfare aspect. Other traits like consumer eating quality, they will crop up in the future, and then at some increasingly further into the future, things like health traits such as worm egg count, EBVs or other things, resistance to certain diseases or conditions will become important as we move further into the future, and our ability to measure them in new innovative ways becomes apparent, they'll come into breeding objectives more and more in the future. But as we sit today, probably the two biggest ones are sustainability, particularly around carbon output, methane production, and dealing with animal welfare in a better way, and things around cow body composition and how they relate to cow welfare are probably the two medium-term things that are really going to increase their presence in breeding objectives.
Perfect. Thanks for that, Brad.
No worries, Emily.
So, Tom, I'm going to direct this question at you. Question is, what is one of the biggest challenges within businesses that could be helped with genetic technology?
Yeah, that's a good question. So from my perspective, one of the biggest roadblocks for genetic improvement, and we know that genetic improvement is linked to profitability. It's a bit like compound interest. It's permanent, it's cumulative, and you can set the direction or your interest rate in the way that you want to. But getting to that point takes some upskilling in terms of knowledge and learning. Basic and applied genetic theory can be daunting for some people. So through projects that I've been involved in, we know that one of the best ways for people to learn and upskill is in groups. So like-minded people getting together and forming groups where they're learning in a group environment where they can share ideas and express that they don't understand some things or need further clarity in learning some particular element of genetic improvement and the theory and how to apply it and how to balance genetic utopia with commercial on-farm practicalities and finding that balance. But yeah, that I would say the biggest barrier is just the upskilling in knowledge and how to apply it.
Great, thanks for that, Tom. That definitely clears up that question, and I think it's a good point you mentioned about producer groups. I think they're definitely valuable. People can learn a lot from others as well. So thank you for that. So David, one of the questions that we had come through was around crossbreeding, and the question was how many generations until hybrid vigour is lost?
So hybrid vigour is generated when you cross two breeds. You can get it within breed, but generally it's when you cross two breeds. So it's hybrid vigour, or heterosis as it's termed scientifically. And so when you initially cross two breeds, you get maximum hybrid vigour in that offspring. And the main reason for that is all our breeds have some level of inbreeding, because that helps determine what is a breed, is that they are closely related. When you cross them, you get recovery from that inbreeding. And so you get increased performance of that crossbred animal above the average of the two parent breeds. So how long does it last? Well, hybrid vigour is only an advantage to the animal itself. So you only generate hybrid vigour in the individual. So if you then take that crossbred animal and back cross it to either one of the parent breeds, you halve the hybrid vigour in the next generation. So really hybrid vigour is lost if you don't continue to crossbreed. So in the first cross, the more diverse the breeds, the more hybrid vigour. So you get more hybrid vigour in a Brahman Angus cross than you will between an Angus and a Hereford. But even in that Brahman Angus cross animal, if you back cross it, then the next generation in that back cross progeny will be half in the progeny, but the mother will still be 100% crossbred. So you can have a crossbred mother and a back cross progeny where half. The only difference is if you develop a composite, you can retain hybrid vigour. And that's where a level of hybrid vigour is exhibited across generations, because you recreate the crossbred. And that occurs if you take two breeds and blend them together. But that only retains hybrid vigour if you manage to avoid inbreeding. So if you're going to start out a new breed and you start with one bull and 20 cows, eventually you're not going to retain hybrid vigour because you can't avoid inbreeding. So successful new composite breeds or synthetic breeds, you need hundreds and hundreds of animals and tens, if not hundreds of sires. In that way, you do retain hybrid vigour in a composite population. And again, in a composite population, the more breeds in the composite, the more hybrid vigour you retain. and the more equal they contribute, the more hybrid vigour. So yes, you do lose hybrid vigour over time if you don't continue to crossbreed.
Okay, that was a really great explanation. Perfect. We actually had a question come in as well, which was somewhat similar in length, so I guess I'll ask you. But the question was, what is a sustainable rate of inbreeding? And I guess you touched on that a little bit, so I might ask you to elaborate a bit more on that.
Yeah, so inbreeding occurs in all populations, and it's a result of animals being related that end up mating. And so in livestock production, obviously we're interested from a performance aspect. And yes, if inbreeding starts to increase, you can get inbreeding depression, particularly in traits to do with fitness, fertility, longevity, survivability. Once you start getting inbreeding, those animals become less fit. And so as a general rule, we try and keep inbreeding below 5%. In some species, they might tolerate 10%, but you start to see inbreeding depression. But the other big issue that you can have with inbreeding is deleterious recessive. So all organisms contain lethal genes, but they're often recessive such that you need 2 copies. So as long as you avoid inbreeding, you don't end up with double copy animals. But the thing with inbreeding is if you start mating highly used bulls that are carrying a lethal recessive and they end up going over their daughters or relatives, then you can end up with 2 copies. Lethal recessives by name are often lethal. And so you have death before birth in terms of fetal losses, but you can also have genetic disorders that are deformities. And that might be, again, terminal, but those animals might be born and they're deformed. And so one of the bigger problems with inbreeding is if you don't avoid inbreeding, then you can multiply up and end up getting lethal recessives in your population. And with modern animal breeding, we can multiply up those animals much more quickly, and then because of technologies like artificial insemination, we can then end up with a population where those frequencies become such that you start seeing the expression of those traits.
Great. I think that summarises the question perfectly. Thanks so much for that, David. All right, so to wrap up the Q&A, I've got one more question. And Tom, I'm going to point the finger at you again. So this final question is, where to next for genomics?
Always pointing the finger at me for the tricky questions. It's a big question, Emily. And from my personal opinion, I would see that a wider, more accessible, rolled out commercial genomic test and individual breeding values available for commercial animals. That's a little bit of a complex space because genomic prediction is driven off a reference population that often it's expensive reference herds or seed stock producers that are supplying data for. So making it at a price point where it's affordable, but we're not stealing business off the people who are doing hard work. In saying that, we do already have some tests available for commercial animals, such as flock profile in sheep, and there are some tests available for cattle as well, both from Australian companies and international companies. But I would see that as a really valuable tool, particularly for people who breed their own bulls or rams or bucks even. So another exciting space that Genomics will be working on is looking at microbiome of the rumens, tying into methane production and rumen efficiency. So that space is really quickly evolving because there's a lot of funding in carbon abatement at the moment. So that will be another space at which genomics will be developing. And then I imagine that one day there will be, say in 15 years' time, a totally different and far more quicker and efficient way to be extracting DNA and getting it into a genetic evaluation. And the way I would liken that is to, in 15 years' time, whatever that process may be, like we think about 15 years ago, the phones were just getting the internet on them. The first iPhone had just come out. And look what we can do now with it. So I imagine that there will be someone or a group of people who will develop something incredible, and it'll be the new norm. But what that is, I don't know, but I'm always excited to see what the future looks like.
Thanks, Tom. I think it's a very interesting space and something that we'll definitely be keeping our eyes on. I think, it's always hard to know in the future how things are going to go, but exciting space. Thanks for that answer. Thank you very much for tuning in for this episode, and more broadly for tuning in with me for the first season of Genetics in the Paddock with Emily. It's been a bit of a roller coaster being the first season, but it's been a great one, and I'm looking forward to starting to film season 2. Your input is always very valuable to us, and I'd love to hear from you guys, especially if you have any questions or suggestions or anyone you'd like to see feature on the podcast. All feedback's welcome, and I'd love to hear from you. Your feedback is what makes our podcast continue to go on. And our aim is always to deliver relevant and information that you find valuable. So thanks again, and we hope to see you soon for season 2.